Today, I am going to talk about the chromosomal aberrations and its implications in human health. Chromosomes are actually kind of thread like structure. When the nucleus in the cell started dividing, the DNA condensed along with histone proteins, and that comes like a thread like structure. That is what we call them as chromosomes. In human, there are 46 chromosomes. 44 chromosomes are common between male and female, but only pair of chromosomes, only 2 chromosomes are different. In case of female, there will be 2 X chromosome, but in case of male, there will be 1 X chromosome and 1 Y chromosome. The Y chromosome is the one which determines the male sex of the individual. So, if there is any change in the chromosome number or structure, it will lead to various diseases, which we will see in detail in the due course of my talk. To detect the chromosomal aberrations, one has to prepare the chromosomes. The best way is take the blood sample, culture them in a media, grow them for 72 hours, and when the cells started dividing, you arrest them with specific chemical, so that the chromosome remains at that particular shape, where we can analyze them and find out the aberrations. To do that, we need to do a specific staining procedure. First, we have to treat the chromosome. Once the chromosomes are fixed and spreaded on the glass slides, it should be treated with the chemical called trypsin, then stained with Giemsa. Once we do that, the chromosomes gives, each chromosome has a specific banding pattern. So, that helps in identifying every pair of chromosome. The chromosome number was not defined in case of human until 1955. There are many scientists who are telling the chromosome number is 48 or 44, but the actual number was identified by Joe Hin Joe in the year 1955. So, that was one of the finding which helped or defined the chromosome number. How do we inherit the chromosome from our parents? As I mentioned, there are 46 chromosomes in every individual, that in case of somatic cells, but in case of germ cell, the 46 chromosome will become 23. Every cell in terms of germ cells like egg and sperm will have only half of it. The 46 chromosomes are in 23 pairs, but in case of sperm and egg, there will be half of it, only 23, not 23 pairs. So, it brings 23 chromosome from father and 23 chromosome from the mother. When the egg and sperm fertilize, then that will become total 46. Chromosomes contain the DNA, which are the genetic material. That is what bring all our characters from our parents. So, coming to the chromosomal aberrations, because any change in the chromosome number or structure, it leads to various disorders. The chromosomal aberrations are broadly classified into two groups. One is a numerical change or aberrations, other one is structural aberration. The numerical aberrations was first identified by Jerome Lejeune and his colleague in the year 1959. Although the Down syndrome was earlier reported with the features, but the extra chromosome number 21 is the real cause for Down syndrome was first identified by Jerome and his colleague in the year 1959. So, that is one of the major finding in case of the chromosomal aberrations. The most common chromosomal aberration in terms of the numerical is Down syndrome. The babies generally born with Down syndrome, basically they will be carrying one additional chromosome 21. They will have growth failure, mental retardation and several other features, and also they are prone for cardiac diseases, and most often they die within the age of 15. So, very rarely the Down syndrome individuals live beyond 15 or 20 years. The Turner syndrome, where there will be one less chromosome, there will be totally 45 chromosome, 44 autosomes will be there, instead of 2 sex chromosome there will be only one X chromosome. And generally the frequency of Turner syndrome is 1 in 2000 to 2500 female birth. The Turner syndrome individual generally will have female phenotype, but they cannot reproduce, the secondary sexual organs all will be very rudimentary. The triple X, we have seen example of Turner syndrome where there is only one X chromosome, and in case of triple X there will be 3 X chromosome, there will not be any Y chromosome. Another example of trisomy is Patau syndrome. It is very rare among the people. The Patau syndrome will have one chromosome with 13 extra, and again the individuals with Patau syndrome may not live for long time. So, Edwards syndrome is another example for trisomy. In this case there will be one additional chromosome number 18. So, this also one of the serious problem, and again these individuals with Edwards syndrome will not survive for long time. The Klinefelter syndrome. In case of Klinefelter syndrome, although the individual with the Klinefelter syndrome may show up with the male phenotype, but they will generally are infertile. They will have tall stature, their mental IQ may be slightly lower than the normal individual, and they will have slightly breast development and other little bit of feminine characters. So, in this case there will be one extra X chromosome, there will be XXY. Although Y is there, because of additional X chromosome, they may not be a perfect male and they cannot reproduce. Now, let us see why this kind of structural numerical abnormality exist? What is the basis for the numerical abnormalities? As I was mentioned, in the somatic cell we have 46 chromosomes. During the production of germ cells like egg and sperm, the chromosome number will become off, 46 chromosome will become 23 chromosome. In case of normal cell division, the cell has to divide and equally the daughter cell will have 23 and 23. But sometime there is a phenomena called non-disjunction. The cell may not, the daughter cell may not have the equal number of chromosomes. Sometime one chromosome can be more in one of the daughter cell and the same thing will be less in other daughter cell. So, in case of the cell become egg or sperm, either with one additional chromosome or one less chromosome, fertilize with the normal egg or sperm, the chromosome number naturally will increase or decrease. And that is being correlated, particularly the Down syndrome, where we see chromosome number 21 is extra in a cell, is associated with the maternal age. Normally, if the mother age is beyond 35 or 40, the chances of getting Down syndrome baby is very high. So, it is advised to have children before the age of 35. The structural abnormalities, we have seen about the numerical abnormalities, where the number in the chromosome change which causes various abnormality and the disease associated with the numerical abnormalities we have seen. So, now let us see about the structural abnormalities. There are several classifications of structural abnormalities. One is called as deletion, where the part of the chromosome will break and go away. Other one is the translocation, where one part of the chromosome will break one chromosome, part of the one chromosome breaks and join with some other chromosome. Inversion, part of the chromosomal material break and go on to join in a different orientation in its homologous chromosome. The ring chromosome, where one single chromosome rejoin after breaking its telomeric region and will become very circular in nature. So, that is what called as ring chromosome. Robertsonian translocation, that is generally associated with the acrocentric chromosome, where the satellites breaks, the two chromosome joins together to make one single chromosome. That is called as Robertsonian translocation. The most frequently associated with mental retardation is called as Fragile X. It causes the break of the long arm of the X chromosome, that generally happens in 1 in 2000 boys and 1 in 4000 girls. So, when there is a fragile X or fragile site, it leads to X linked mental retardation. In general, X linked disease are mostly affected by the males, because there is only one X chromosome in the male, if that has abnormality, and the phenotype will show up in the males, but in the females mostly they all will be carrier because of 2 X chromosome, unless otherwise if it is the mutation or the abnormalities in both the chromosomes, it may not show up the disease. The Fragile X, to look for the Fragile X, there is a specific technique available. One has to grow the cells, blood cells, in a medium which contain specific chemical, and one has to prepare the chromosome as I described earlier, treat with trypsin, stain with Giemsa, then you see a small fragment just hanging at the end of the long arm of the X chromosome, that is called as fragile sites. So, that in molecular level, if you look at, there is expansion of CGG repeats. So, that leads to severe form of mental retardation. In case of female, generally there will be a carrier, and the males are severely affected with the Fragile X syndrome. The structural rearrangements of the chromosomes can be classified into balanced translocation and unbalanced translocation. Balanced one are like inversion, reciprocal translocation, and Robertsonian translocation. The unbalanced one are deletion, duplication, ring chromosome, and isochromosome. The unbalanced rearrangements generally is loss or gain of chromosomal materials and can show in different types, which I described like translocation, inversion, deletion, and duplication. And of course, these are all associated with different phenotype, that means different diseases. As is shown here, the deletions are at either in the tip of the chromosome or somewhere in the middle of the chromosomes. It breaks and the material goes away from the chromosomes. So, generally in that case, you will see out of 2 pair, 1 pair is either short, because it is a deletion we are talking about. So, naturally one of the chromosome will be shorter than the other chromosome. So, the deletions are detected at microscope level. That is fine. The sum of the deletions cannot be detected at microscope level. They are called submicroscope deletions. So, that can be evaluated or studied by DNA level or molecular level. There are few examples which I wanted to discuss, like cry-to-chat syndrome. So, in that case, there will be deletion of the chromosome number 5 in the P arm. Another example is deletion of long arm of chromosome number 18. So, this will lead to severe form of mental retardation. I mentioned about Cray-Duchat syndrome. It is a partial deletion of chromosome number 5, and the frequency was found to be 1 in 50,000 live birth. The patients will have round face, and most often the individuals will be with mental retardation. This is also example of chromosome number 22 long arm deletion. Now, we come to chromosomal duplications. As is shown here, part of chromosomal material you see is duplicated. This duplication we often see in the region of non-coding sequences. Particularly if you take chromosome number Y, there are some genes with 37 copies. Of course, most of the copies are not functional. So, in case of Y chromosome, if there is a duplications, particularly in the non-coding sequences, some regions multiplied or duplicated for thousands of times. So, that does not affect, because it is a non-coding region, that does not affect the individual, but the duplication of chromosome material in the coding region that affects the phenotype of the individual. That means, it causes diseases, various diseases. Another one is the insertion. It is a delete and gain additional chromosome material. It is called as insertion. There are few examples for chromosomal duplications, which again leads to various diseases. I also mentioned about chromosome inversions, where part of chromosome material deletes and invert and rejoin in the chromosome. The another example is reciprocal translocation. Also, we have balanced reciprocal translocations. There is a deletion and translocation. There are few examples within the chromosome X. There is a deletion and translocation in chromosome number 4 and 6. This is also commonly known chromosome abnormalities, the Philadelphia chromosome, which involves chromosome 9 and 22, which is associated with cancer. There is also another abnormality called as isochromosome. Normally, the chromosomes are divided vertically. In case of isochromosome, the chromosome divides horizontally. The same arm, whether short arm or long arm, will become double. So, we have, rather than have P arm and Q arm will have one arm with two copies, it is called as isochromosome. I also mentioned about ring chromosome, where both end of the chromosome breaks and the ends, broken ends rejoins, give rise to the ring chromosome, which are circular in shape. There is also other abnormality called as micro deletion, that is mostly seen in case of Y chromosome. The Y chromosome, there are three regions which are responsible for sperm production, azoospermic factor A, B and C. If there is any deletion in those regions, it ultimately leads to abnormal semen profile. So, that is associated with male infertility. So, this is an example which shows where there can be a deletion and duplication in sex chromosome, which again leads to infertility and ambiguous genitalia. So, coming to the treatment part of the disease, obviously there is no treatment for the genetic disease, because the every chromosome or every cell having the particular abnormalities. Although the stem cell therapy, gene therapy are being initiated, but there is no clear cut treatment for the genetic diseases, particularly the chromosomal associated diseases. The important aspect of the chromosomal aberration is counseling the parents. If either mother or father carry the chromosomal abnormality, of course, most of the chromosomal abnormality leads to infertility or recurrent pregnancy loss. Some can be transmitted from one generation to other generation. So, those can be evaluated, assessed in the prenatal stage, then we can provide counseling to the family to avoid such abnormality in the coming generation. So, with that I conclude my talk. I hope you all would have enjoyed in listening this lecture. Thank you very much.